Hi everyone, welcome to Hall. This is Vidyam Academy's NEET Initiative Most Expected Question Series. This is Rajiv Patel. Today's topic is 12th Standard NCERT Biology. The first chapter, Sexual Reproduction in Flowering Plants. Here the subtopics are Endosperm, Embryo, Seed, Apomixis and Polyembryony. Let's welcome to all. Let's get started. The first question is, in nuclear endosperm, the first division of the primary endosperm nucleus, famously known as pen, is option A, followed immediately by cell wall formation. Option B, meiotic producing haploid nuclei. Option C, mitotic with no cell wall formation initially. Option D, followed by formation of aleurone layer. So what is the correct answer of this question? Would you guess, anyone? Your time starts now. Yes, the answer is option C. Once again the question. In nuclear endosperm, the first division of the primary endosperm nucleus is. Here the explanation. Why C is absolutely right answer here. Mitotic with no cell wall formation initially. We need to see how. Nuclear endosperm is the most common type of endosperm development in angiosperms. When primary endosperm nucleus, famously known as pen, formed by fusion of one sperm with the two polar nuclei, begins to divide, it does so by repeated mitotic division without forming cell walls between the daughter nuclei. This results in a multinucleate, koinocytic mass of the cytoplasma with many free nuclei. Cell walls forms only later, typically from the periphery towards the center. The classic example is coconut. Coconut water is the liquid phase, free nucleus stage, and the white solid kernel is the later cellular phase. This is the explanation why nuclear endosperm, the first division of primary endosperm nucleus pen, is mitotic with no cell wall formation initially. Then next, let's move to question number 2. So it's an application based question. You can easily identify what is the answer. A botanist observes a seed in which the endosperm is completely absent in the mature stage. Yet cotyledons are thick and fleshy. The seeds most likely belongs to option A, maize, option B, castor, option C, pea, option D, wheat. What is the answer for this question? Would you guess? Your time starts now. Yes, the answer is option C. Why and how? Let's go to explanation. Here, seeds are classified as endospermic, albinous, retaining endosperm at maturity, or non-endospermic. Once again, you need to understand why P is the right answer here. So here, endospermic, albuminous, that is very important. So which we are hearing explained. The endosperm completely absorbed by the cotyledons during seed development. In P, a pisum sativum, endosperm is formed after double fertilization, but is progressively absorbed by the developing embryos cotyledons, which become thick, fleshy nutrient storage organs. So at seed maturity, no endosperm is visible. The cotyledons are the food store. That's what option C, P is the right answer, which question demands. And next, move to question number 3. It's a diagram based and conceptual understanding based question here. In the embryo of a dicot, the structure that connects with the embryonal axis to the cotyledons and also serves as the first photosynthetic organ in some of seedings is. So this is the question. Option A, hypocotyl. Option B, epicotyl. Option C, scutellum. Option D, culiopetal. So what is the answer for this? Option 4, which we have given under here. Would you guess? The answer is option A. Why it is the right answer? In a dicot embryo, the embryonal axis has two regions. One is hypocotyl, below the cotyledons, above the radicle, and the epicotyl above the cotyledons, below the pluminal. So here, the hypocotyl directly connects to the point of cotyledon attachment during germination in epiphyseal seedings. For example, bean. The hypocotyl elongates and pushes the cotyledons above ground. Since the cotyledons are briefly exposed to sunlight in such cases, the hypocotyl cotyledons region may be the first to turn green and photosynthesize in some species. That's what. So here the answer A. Hypocotyl is the exact answer of this question. And next move to question number 4. It is assertion and reason type. Assertion A. Coconut water is a nutritive liquid endosperm consumed by the developing embryo. This is the assertion. Again, reason we have coconut endosperm is of the allobial type and remains liquid throughout seed development. Option A. Both A and R are true and R is the correct explanation of A. Option B. Both A and R are true and R is not the correct explanation of A. And C. A is true but R is false. And finally D. A is false but R is true. What is the right answer for this? The correct answer is option C. Here the explanation why assertion is true and reason is false. So here we have coconut water. Coconut water, tender coconut liquid is indeed the liquid nuclear endosperm. It contains sugars, amino acids, and growth hormones and minerals that nourishes the developing embryo. Hence, nutritive liquid endosperm is correct. But R is false. Do you know why? Here the explanation. Coconut endosperm is of the nuclear type, not allobial. So in coconut, the pen undergoes free nuclear divisions. Nuclear endosperm. The early stage is liquid coconut water is equal to free nuclei in fluid. And the later stage is cellular, white solid kernel. Allobial endosperm is a characteristic of monocots like orchids. The first division produces two unequal chambers. So the reason incorrectly identify the type, making the answer. That's what A is absolutely true, but R is false. That's what option C is right answer here. And next move to question number 5. Match the following seeds with their connect endosperm type. Column 1 is a plant. Column 2 endosperm type. Option 1 coconut. Option 2 orchid. Option C petunia. Option 4 pea. So here one side is the options. Coconut, orchid, petunia and pea. So here column 2 cellular, nuclear, absent non-endospermic and allobial. What are the answer? Option A 1B, 2D, 3A, 4C. Option B 1A, 2B, 3D, 4C. Option C 1B, 2A, 3D, 4C. Option D 1D, 2B, 3A, 4C. What is the answer? Would you guess? Your time starts now. Yes, the answer is option A. Match the following seeds with their correct endosperm type. So 1B correctly matched, 2D correctly matched, 3A correctly matched, and 4C correctly matched. So here once again, how and why. Coconut, it's a nuclear. Pen undergoes free nuclear division, liquid and solid endosperm. Nuclear type. That's what. So it's correctly matched. Orchid, allobial. Orchid show allobial endosperm. First division divides pen into a large chalgel and small micropillar chamber. So that's what it's correctly matched. Orchid allobial. Petnia, it's correctly matched with the cellular. Every nuclear division in petnia endosperm is immediately followed by the cell wall formation, cellular endosperm. That's what it's also correctly matched. And the fourth one, P, here the absent non-endospermic, because P is X albuminous endosperm observed by the catalydents and during development. That's what these are correctly matched and remaining unmatched. That's what option A is correct answer in this question. And next move to question number 6. Which of the following statements about Apomixis are correct? Yes, the first one, it produces seeds without fertilization. Option 2, it always involves meiosis. It is commonly seen in Asteraceae and some grasses. Option 4, hybrid Viga can be maintained across generation using Apomixis. What are the answer? Which of the following statement above Apomixis are correct here? The first A option, 1, 2, 3 only. Option B, 1, 3, 4 only. Option C, 2 and 4 only. Option D, 1 and 3 only. What is the answer? Would you guess? Yes, the answer is B. So here statement 1, 3, 4 are correct. The subject is concerned Apomixis. So here the statement 1 is correct because Apomixis is asexual seed formation. Seeds develop without fertilization. Embryos form from diploid cells. So here nuclear cells, unfertilized eggs and integument cells, which works and which comes under statement 1. Statement 3 are correct because Apomixis is commonly found in family Asteraceae and grasses, as well as in citrus and mango, even 2. Option 4 also correct because since Apomixis produces seeds without fertilization and without segregation, hybrid characters are clonally preserved across generations. This is the greatest agricultural significance. By the way, so statement 2 is incorrect because Apomixis bypasses meiosis. No reduction division occurs. The megaspore mother cell either skips meiosis or diploid sporophytic cells directly from the embryo. So that's what statement 2 is absolutely wrong, and 1, 3, 4 are right here. Next, question number 7. A plant breeder successfully develops a high yield hybrid variety to maintain hybrid character generation after generation without repeated crossing. The most ideal biological strategy would be what? Option A, induced polyploidy using colchicine. Option B, vegetative propagation only. Introduction of Apomixis into the hybrid. Option D, cleistogamy. So here the options are. What is the right answer? Yes, the right answer is option C. Option C, introduction of Apomixis into the hybrid. Why and how? Let's see. When a hybrid is produced by crossing two inbred lines. So here, hybrid heterosis. If this hybrid is allowed to the reproduction sexually, Mendelian segregation breaks up the favorably allele combinations. Hybrid vigor is lost. Repeated crossing is expensive and time consuming. If Apomixis genes are introduced into the hybrid, seeds are produced asexually without fertilization. The embryo is a genetic clone of the mother plant. No segregation occurs. So hybrid characters and hybrid vigor are faithfully transmitted to every seed. Generation after generation without any crossing. This is the prime agriculture goal of Apomixis research. That's what here colchicine and allele are most important keywords in this question. The next question is conceptual understanding question once again. Which of the following correctly distinguishes adventive polyembryony from cleavage polyembryony? Here the options are. Option A, in adventive polyembryony, extra embryo arises from the zygote splitting. In cleavage polyembryony, they arise from a nucellar tissue. Option B, In adventive polyembryony, extra embryos develop from nuclear integument cells. In cleavage polyembryony, the zygote or proembryo splits. Option C, both involves tissues outside the embryo sac. Option D, adventive polyembryony is a type of apomixis, which cleavage polyembryony is sexual. What is the answer for this question? Do you guess the right answer? The correct answer is option B. Why and how? Let's see. Polyembryony, so more than one embryo in a seed. Adventive polyembryony, extra embryo arises from sporophytic cells. Specifically, nuclear cells are integument cells that proliferate and develop into embryos independently, alongside the zygotic embryo. These cells are diploid and no fertilization occurs. This is a form of apomixis, nuclear embryony. It's a classic example. If you want to give here, it is citrus, lemon, orange. It's a nuclear embryony. So even mango. So cleavage polyembryony. So here we need to understand the answer is the zygote itself or the early proembryo splits into two or more units, each developing into a separate embryo. This is a sexual process. Fertilization has occurred. Let's say, for example, nymphia. So water lily are orchids. And next move to very important question. Question number 9. In citrus, when seeds are germinated, multiple seedlings emerge from a single seed. This happens because option A, multiple egg cells are fertilized simultaneously. Option B, synergies develop into the embryo due to parthenogenesis. Option C, nucellar cells proliferate and form embryo alongside the zygotic embryo. Option D, endosperm nuclei directly differentiate into embryos. What is the answer for this question? Do you guess? Option C. Why? It is the very best and classic example here of the citrus. It's an adventive polyembryony, nuclear embryony. The nucellus is the maternal sporophytic tissue that surrounds the embryo sac. In citrus, diploid nuclear cells are stimulated to proliferate and develop directly into the embryos without any fertilization. These nucellar embryos developed alongside the zygotic embryo within the seed. When the germinated multiple seedling emerge, some from the single zygote embryo sexually produced and many from nucellar embryos asexually produced, genetically identically to the mother. This is exploited in horticulture. Nucellar seedlings are disease-free, true to type rootstocks. That's what this is the explanation for this question. And final question of this session, consider the following events after double fertilization. 1. Statement, one sperm fuses with the egg zygote. And 2. One sperm fuses with the two polar nuclei, primary endosperm nucleus pen. Option 3. Pen undergoes free nuclear divisions. Option 4. Endosperm is consumed. The seed becomes non-endospermic. So which sequences are correctly or describes the development of the seed like P? Option A, 1, 2, 3, seed retains endosperm. Option B, 1, 2, 3, 4, cattyledons become thick. Option C, 2, 1, 4, 3. Option D, 1, 3, 2, 4. What is the answer for this question? Would you guess? The right answer is option B. So here, 1, 2, 3, 4. Cattyledons become thick. So these are correctly describes the development of C's like P. So 1, 2, 3, 4 exactly matched. Because the step 1 first sperm fuses with the egg cell, diploid is zygote 2N. So this is syngamy. Step 2, second sperm fuses with the two polar nuclei, primary endosperm nucleus pen, which is diploid 3N. Both fusions occur simultaneously, double fertilization. Step 3, pen undergoes nuclear free nuclear divisions. Endosperm begins to form around the developing embryo. So final step 4, as the embryo develops, cattyledons absorb all the endosperm, speed becomes non-endospermic. Cattylids become thick and fleshy. The mature pea seed has no free endosperm. That's what this the steps which are 1, 2, 3, 4 exactly describe the statement. That's what answer B is right answer in this question. So here we explained 10 most important and expected questions for your upcoming NEET examinations. And next, I will come up with another topic which are very important for your NEET examination. Till so far, I would like to conclude this session with very well goodness. Thank you and thank you so much to all.